Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. This is the second video on the series of innate immunity, and in this video, I shall talk about structure of TLR or Toll Like Receptor. So, in short, in this video, I shall brief you about the physical structure of TLR and its function. We start this talk with Professor Bruce Beutler's walk with his father. When Professor Ernst Beutler, the senior Beutler, what is called as his father, initially it is always said the parents are the best teacher. So Bruce was a small kid, he was holding his father's hand, and once he was holding his father's hand and he was walking through the groove of redwoods in Sequoia National Park, US. And then small kid Bruce asked a question to his father, why is it that trees simply do not rot? If you don't keep the vegetable inside the refrigerator, it rots, it putrefies. And he asked the question, why certain trees simply do not rot? Entire life they remain as it is, fresh. Why they don't rot? That was the question which Professor Bruce asked his father. And in fact, that question in childhood days led Bruce to win a Nobel Prize when he became a scientist and an adult. So that is how the first, good science is not about the equipment, facilities or infrastructure, it's all about the right question. So that was the question which Bruce asked during the childhood. As time progressed, soon Bruce joins academics, and after joining academics, what might be the nature of contact between the microbe and the host was the modified question which Bruce asked. What might be the nature of contact between the microbe and the host? How microbe recognizes host? How host respond to microbe? These were some of the questions which Professor Bruce kept asking until he found the answer. So these two question and answer sessions led to a fantastic Nobel Prize in Physiology and Medicine. We shall have a small bit of history into that aspect in this talk. So what is the history of TLRs? It all began in a 2014 paper. Of course, the Nobel Prize was much earlier, but then in 2014 paper, I have the paper for you. It was published in Frontiers of Immunology. It was a review article which talked for the first time the Toll Like Receptor signaling pathway. This was many years after the Nobel Prize was awarded. This is a fantastic review. You can go through the review to get a gist of the entire pathway without referring some 15 or 20 papers. Rather, you can go through this review paper. So, Frontiers in Immunology published the Toll Like Receptor signaling pathway, and a part of the history I have taken from this paper. The Nobel Prize oration lecture, when he was asked to address the audience, what Professor Bruce Beutler chose was how mammals sense infection. How mammals sense infection from endotoxin to the toll-like receptor. He did his piece of work from Center of Genetics of Host Defense, Southwestern Medical Center, Dallas in Texas. This seminal work led to the award of Nobel Prize to Professor Jules Hoffmann 2011 and Bruno Lemaitre, who did a seminal work in 1996. It's a combined work of everyone that led to Professor Jules Hoffmann winning the Nobel Prize in 2011. So, what are the experiments? The group observed or experimented with mutant flies, mutant Drosophila was their experimental model. What they observed, when there was a mutation in Drosophila, the common fruit fly, there was a mutation in Toll. All those flies that had mutation in Toll, those flies were susceptible to lethal infection with a fungus, Aspergillus fumigatus. On the contrary, the wild type flies, where there was no mutation in Toll, those flies were not susceptible to immune or lethal infection with Aspergillus fumigatus. So, quite obvious, they reason when mutation in Toll is there, the flies do not become susceptible, whereas no mutation, the flies are not susceptible. So, these two aspects lead to a fundamental understanding came in the mind of the scientific group that something might be there in this Toll which can confer disease resistance, or it can prevent the susceptibility to infection. Then came the year 1997. Author of the famous book Immunobiology, always we say Janeway and Travers. So, this is Professor Charles Janeway together with Ruslan Medzitov. This was a piece of work in Russia, that was 1997, and in 1997 they came up with this paper, the Toll Like Receptors Signaling Pathways, in a special section that got published later in 2003, the work of 1997. So, Toll Like Receptor Signaling Pathways. What this group, that is Medzitov and Charles Janeway, observed, there were certain human proteins that were identified by homology in its cytosolic domain and that of Toll. What they did, they activated the expression of the immune response gene into the transfected human cell line. After the transfected human cell line experiments, they named this as a Toll, that is TLR4. So, that was the first TLR they experimented with, that is TLR4. Things were not that clear at that point of time too. It was in 1998. I know my lecture I started with Professor Bruce asking his father that why the trees do not rot. So, it was in 1998 that Professor Bruce Beutler also worked on this, and together with Professor Jules Hoffmann, he was awarded the Nobel Prize in Physiology and Medicine in the year 2011 for his seminal work on innate immunity, especially the TLR signaling. So, what was Professor Beutler's contribution? Mice homozygous for LPS locus, he saw, was resistant to lipopolysaccharide LPS. molecular biology was available. They did DNA sequencing. The mouse LPS gene encoded a mutant form of Toll like receptor, that was TLR4. This TLR4 differed in single amino acid, just one amino acid, and you change one amino acid, the nature changes. Therefore, Professor Beutler concluded that TLR4 is indispensable for the recognition of lipopolysaccharide. If you see their Nobel Prize lecture or their question, how a host recognizes a pathogen, one of the ways Toll like receptors. So, TLR4 is indispensable for the recognition of gram negative bacteria's lipopolysaccharide. So, that was Professor Beutler. Till now, that is 26th of March 2021 that I am talking to you, as of now there are 11 TLRs known in humans, and in mice there are 12 TLRs that are known. This is the data so far. Now we go to the structure of TLR. This is the structure of TLR. We have this is cytosolic domain, this is TM transmembrane domain, and this is EC, that is extracellular domain. This much is extracellular, this much is transmembrane, and everything below the transmembrane is cytosolic domain. So, extracellular domain, transmembrane domain, and cytosolic domain. Remember students, the extracellular domain consists of LRRs, that is leucine rich repeats, that is the exterior domain or extracellular domain. This is a ribbon model of the exterior surface. You have repeated rich repeats of leucine rich repeat, that is LRRs. Overall the structure is like a hairpin loop, you can see. Then you have the transmembrane domain and the cytosolic domain. The cytosolic domain contains 3 boxes, box 1, box 2 and box 3. These 3 boxes contain conserved sequences, because a TIR or Toll like receptor is evolutionarily conserved. It is found in across all phylogenetic lineage, right from the plants to the higher organism. So, TIR domain, and TIR stands for Toll interleukin receptor, that is TIR, that is Toll interleukin receptor. You have homology sequence, that is of box 1, box 2 and box 3. Snapshot summary of what I taught, because 2-mark question is often asked. Describe the structure of Toll like receptor. You have to draw a small schematic figure, not very artistic figure, and explain the key points. So, this is the Toll like receptor. On the extracellular domain, you have repeating segment of 24 to 29 amino acid. These 24 to 29 amino acid contains the leucine rich repeats, that is LRRs. All TLRs contain leucine rich repeat, and a subset of LRRs that makes the extracellular ligand binding domain of TLR. Then you have the Toll interleukin 1 receptor, that is 3 regions that is highly conserved among the members of TIR family. So, that is in the cytosolic domain that I showed in the earlier diagram. And finally, you have box 1, box 2 and box 3 evolutionary binding sites of intracellular proteins. They participate in signal transduction pathway. That of course will be in the next segment of my talk, and that contains the box 1, box 2 and box 3 is evolutionary conserved. These boxes are present in the TLR domain. Earlier it was known as Toll interior domain, but now it is known as Toll interleukin 1 receptor domain because of the serial homology between the two structures. The next figure that I am going to show has undergone several modifications, because if you talk about the current institute where I teach, right from your JS's batch, when the chapter was not existing, even innate immunity as a chapter was not there. If you today also, if you check 5th edition of Kubi, the chapter is missing. The next batch, that is your Manthan sir's batch and Chandni madam's batch, that was the batch when the innate immunity as a chapter came up for the first time. And I still remember one student from that batch asked a very good question, that why initially in that batch, I will show the figure, right. One of the days I will bring the old books figure also. And in that book, that is the 6th edition, all figures were single, that is homodimer. Everything was single. Another thing that was known at that time, you know what you can see? Tarak Mehta ka ulta chashma you can see over here. This part, the TLR is in the interior part, whereas in the next diagram, the TLR part is in the exterior domain. In that particular book, the 6th edition, when I taught in 2009, all figures were in the interior domain. And this, students, what it suggests, how science progresses. Now that we are studying the same chapter, the same topic, right, from 2009 to 2021, 7 round times the figure changed in my PPTs. So now if you see the figure, I will get an eraser. Now if you see the figure, you have endosomes and lysosomes. The TLR is in the exterior, whereas in the plasma membrane, the TLR is in the interior. Very interesting, students. You all know viruses do not thrive outside the host cell. So viruses are endogenous. Whenever you have a virus invading your cell, virus is always inside the cell. So automatically the viral signaling will be from inside, and that is why the domain, the leucine rich repeat in case of endosomes and lysosomes is inside. The same thing when it comes to an extracellular bacteria. Extracellular bacteria, as the name suggests, the bacteria comes from outside. So obviously the domain, the LRR domain is outside. In other words, what I am trying to say, why this field won Nobel Prize? Because TLRs or toll like receptor can take care of all specific infection, be it bacteria, both extracellular as well as intracellular, fungi, the viruses, always they are endogenous, as well as parasite. So this is your Kapil Dev or Imran Khan of cricket, all-rounder, that all across the innate immunity, toll like receptor has a role, and toll like receptor is also like your Rameshwaram bridge, which is the connecting link between your innate and adaptive immunity. So all these background information, and I am not repeating this 4, 2, 1 and all those, because I have a separate entire lecture dedicated for this. Only thing I would like to highlight is that the lipopeptide is in the toll interior domain, that is TLR1 and TLR2. Another thing I would like to highlight before I go forward, student, please note, if you see TLR4, TLR4 is found in the endosome lysosome, TLR4 is also found in the plasma membrane. So, it is a dual purpose versatile TLR. Now, we move forward to the types of TLRs. What are the types of TLRs? What are the types of TLRs? We have the first is TLR 1, 2 and 6. These are theoretical aspects. So, I shall be a bit fast. TLR 1, 2 and 6, they recognize lipoteichoic acid from gram positive organism and lipoarabinomannan from mycobacterium. So, someone asks you an MCQ in competitive examination, which TLR is responsible for recognition of tuberculosis, your answer should be 1, 2 and 6. Why? Because those TLRs have the binding site for lipoarabinomannan found in mycobacterium or tuberculosis causing organism. Coming to TLR2, it is a specific which recognizes lipopolysaccharide and it cooperates with TLR1 and 6. TLR1 and 6 associate with TLR2 to discriminate diacyl and triacyl tripeptide or lipopeptide. So, that was about TLR1, 2 and 6. Further, we cannot forget the role of macrophages. Monocyte, when it invades to tissue, become macrophage. Macrophages from TLR6 deficient mice did not produce CD36. So, experimental proof that TLR6 is important for expression of cluster of differentiation 36 on the surface. Similar for triacyl lipopeptide, and again experimental evidence, TLR1 deficient mice reacted with diacyl but not with triacyl lipopeptide. So, there is a distinction between di and tri, and always there is a correlation with TLR6 between the recognition aspects of diacyl and triacyl lipopeptide. Coming to TLR3, I have highlighted TLR3. TLR3 recognizes double stranded DNA during the viral replication and type 1 interferon is produced. TLR4, as I suggested, I showed earlier, it is both endosomal as well as in plasma membrane. It is LPS receptor and transducer signals. LPS LBP complex is formed associated with CD14. MD2 is the secreted protein which reacts with extracellular region of TLR4. These aspects are not found in book. You can refer to the review paper, those who are interested. Otherwise, these exclusive details won't be asked in exam. But yes, for being in masters, you should be in a position to know all this. MD2 is the secreted protein that reacts with extracellular region of TLR4. Extracellular region is the TLR that is present in the plasma membrane. TLR5, it recognizes bacterial flagellin as a ligand. TLR7, it used the treatment of infectious disease. It is used as an antiviral imidazoquinolone group of drugs that are used against human papilloma virus, HPV. Now, of course, all children who are born after 1995, they are given HPV vaccine. If it's a female child, they are given human papilloma virus vaccine. There is a strong correlation with first semester and second semester. That's in a joking term. First semester in cell biology, we studied cancer. There is a strong correlation with human papilloma virus to the cervical cancer risk in females. So, that is recognized by TLR7. TLR9, it recognizes bacterial viral DNA as well as your malaria ka janam data, what is called as the plasmodium of malaria. I just remember TLR9 recognizes bacterial viral DNA and plasmodium. It recognizes CpG DNA. It is not a cell surface receptor unlike others, which indicates a different mechanism, that is for TLR9. Finally, TLR10 and TLR11. TLR10 has anti-inflammatory reaction. TLR10 has anti-inflammatory reaction unlike other TLRs. It is more helpful in suppressing cytokines. So, whenever, just like COVID-19, when you have a cytokine storm, you want to control the excess influx of cytokine, you can always play with Toll Like Receptor 10, because out of all TLRs, it is TLR10 which is anti-inflammatory in nature. TLR10 mechanism was found to suppress, how it suppresses inflammation by blocking NFKKB and MAP kinase pathway. These two pathways are very important in culminating the result of inflammation. TLR11, it is present in monocytes, macrophages and dendritic cells. Whenever you have an infection of Toxoplasma gonadii, that is STD, sexually transmitted disease, it occurs in the form of ligand is profilin, and it prompts the dendritic cells for proliferation purpose. I am not going into the detail. If you refer your book, whatever I have taught, this is given in 2-page summary, the Toll Like receptor and the microbial ligand, all the 11 TLRs that I have taught. This is given in 2-page table together with the ligand and the microbe. I am not repeating it because I have just talked about all this. Just now I talked about TLR12, that is profilin, Toxoplasma gondii, and TLR13, ribosomal RNA is unknown. It is in gram-positive bacteria and vesicular stomatitis virus, VSV. When you talk about the TLR signaling pathway, there are two key adapters, and I am just coming to the last part of my talk. I would request Megha madam to just pass on the attendance link to you people, right? So that side by side you can fill your attendance. Megha, you can just pass on the attendance. TRIF, another adapter is TRIF, TIR domain containing adapter, that is interferon beta factor, and TLR3 is TRIF binding, whereas TLR4 uses both pathways, both adapters, TRIF as well as MyD88 and TRIF. So, whenever you talk about TLR signaling, that is in next lecture, we have two signaling pathways. One is MyD88 dependent pathway, another is TRIF dependent pathway. Whereas TLR4 is very rare because TLR4 uses both pathway, that is MyD88 dependent pathway as well as TRIF dependent pathway. There are additional adapters. One adapter is TRAP. TRAP stands for TIR domain containing adapter protein. It recruits MyD88 to TLR2, 6 and 4. TRAM stands for TRIF related adapter molecule. It recruits TRIF to both TLR3 and endosomal lysosomal TLR4. There are adapters of TLR pathway. MyD88 is the adapter used for most TLRs. TRIF is uniquely associated, MCQ question, very important MCQ. Which are the TLRs which utilizes TRIP pathway? One is TLR3, another is TLR4, very easy to remember, only two, and they are localized to endosomes. Apart from these two, all others use MyD88, that is used by most TLRs. Except for 3 and 4, all other TLRs use MyD88 dependent pathway. When you talk about the signaling from interleukin 1 receptor, binding, ligand binding, activation of MyD88 pathway, you have ubiquitination reaction, docking proteins, activation of MAP kinase pathway and NFKB pathway. I have purposefully not elaborated the pathway because that comes in my next lecture series, where I will be talking about the entire signal transduction pathway. It is a lengthy lecture. So, finally, I would like to end the presentation on the structure of the toll like receptor.